Good evening, everyone. And we are going to discuss about an interesting topic, and it is part of third unit. So it is interesting for several reasons. And today I will just discuss a few things about cancer. And the topic as such is about cancer stem cells. Okay. So I would hopefully I will discuss these two or mostly about cancer basics. It's the last hour, so let's see. I want to know how many of you have already are you doing cancer biology elective? Have you done or you haven't done?
It's parallel to this, so
Oh,
we
so
couldn't.
you opted this one, the stem cell biology course instead. Okay. So in that case, I would focus a little bit on cancer basics and give my perspective on it, and then that will be the common in majority of the other things about cancer. Let's check let's try to discuss about cancer. Cancer is about abnormal cell growth, or you call it as neoplasia. That means a cell is has two fates. And the general model, if you remember, and we will also discuss more on that in this case, in today's lecture, is that cell has to divide when it should. That means the cell division is highly regulated. And eventually, after division over time, and there will be wear and tear. And during that time, because of during the wear and tear, what also happens is that the cell will die out. That is a common scheme for majority of the tissues. So when the cell should divide, this part of it is highly controlled. Okay. And that is by cell cycle regulation. And when we have any cell that is out of the cell cycle regulation and dividing uncontrollably, and that is abnormal cell growth, because of which the tissue also could grow, and that is cancer. There are two types, malignant and benign. Benign is just growth and it is not so troublesome, or it does not cause so much problem to the human body. The human body has so many tissues and organs, and all of them should work in coordination with each other based on the resources the individual has, the amount of food to start with. Then if one of the tissue is growing large in number, then it is a problem to the whole system. The system here is the human, because there will be the coordination between the tissues will spoil and then will be affected. Moreover, the growing cell mass will have its own demands of energy and space, and say blood supplies the energy, oxygen, and so on, right? That means if one tissue is occupying more of it, then it means it is causing trouble to the human in general, in total. Same way, there is one more problem is these tissues, when the cancer, the tumor, when it is growing and occupying space of the adjacent tissues, it is going to interfere with the functions of the other tissues. That is another problem. And in the tissue where the the tumor is growing, there itself the tissue might not be as functional. Function is compromised, and that is going to cause a problem for the individual, because slowly the energy, as the tumor is growing, it will have more and more demands for space, energy, oxygen, and all. So it will slowly take away the resources of the body, and then it will keep on growing, interfering with multiple organs, and then eventually killing. So there are several tissue types in our body, and many of the tissue types are prone to becoming cancerous. So the benign ones are just mass of growth, and they do not do anything. They grow much slower. That is what they are, slow in growth. They do not invade the other tissues, and they do not circulate to the other parts of the body. Whereas malignant ones are the problematic, where they grow rapidly and they are much more aggressive, and they move into the surrounding tissues and eventually move through the blood system, move into the whole body, and then establish secondary tumors in different other locations. Cancer is a genetic disease in the sense that it is caused because of mutations. And when you say genetic disease, we usually think of it as it should be inherited from one generation to the next generation. It may not be true that way, but if there is a mutation in one cell, it will be inherited by the daughter cells, at least in that sense. Although these tissues may belong to the somatoplasm, still it's a kind of genetic disease because mutations happen in the DNA, and that is the cause for the cancer. I would want you to also have a different perspective. If you remember molecular biology, and if we discussed about, we had a whole chapter on mutations. We know that mutations can be caused by agents, which you can call it as induced mutations, like radiation, physical and chemical ones, like several other chemical compounds can also cause mutations. There is another set, if you remember, which are spontaneous. That means it's not about some mutagen coming and inducing mutations, but they happen within the body just like that, without any outside influence. There are two important things that you have to look at. One of it is replication errors, right? Every time there is replication, because DNA polymerase does make 1 in 10,000 or 1 lakh bases polymerize, there is a mistake possible. So there are errors, and we also remember, I hope you can recollect that we have mutase and this replication error. The mismatch repair mechanisms are there to clean up all the errors made by the during replication. And still some of them will escape these and then they become as mutations. They will be established as mutations. There are others because of oxidative stress and several other stresses, hydrolytic reactions, some of the bases losing, and they can also be established. They will be tried. There will always be an attempt to repair the damage, okay, by the repair mechanisms. But some do escape these repair mechanisms and establish themselves as mutations. So UV light and so on can induce, if we think about bleomycin, other things, as well as ethylium bromide. I'm not too sure how potent is ethylium bromide in mutagenesis, but some people do say that it is. It could be as well. So it is about mutations, and that is why we do describe cancer as a genetic disease. The second thing is some people describe cancer as immunity. If we consider that spontaneous mutations are happening all the time, and we have a individual, and the individual, within the body of the individual, there are cells always constantly arising, which are mutants. And some of the mutants could be cancer. That means they can go uncontrollably. But then there is one point, is that immunity. If you're reading immunology now, you will probably come across it. Immunity is something that is inherent in our body that protects the body from pathogens that come from external side, but also protects all the from the mutant cells, right? So it is assumed that the mutants in our cell, in our body, are constantly happening. Mutations are constantly happening and mutants are produced. But it is the immunity of the body that will kill these cells. So therefore, there would not be any cancer. But if there is any decrease in immunity because of a disease or a physiological or physical state, or it could be because of medicines, in those situations, there is a high incidence of cancer. There is a correlation, right? That if there is immunity it is something like that. Immunity is inversely proportional to the occurrence cancer incidence, right? If you also see old people in whom the immunity is likely getting lower, the lesser the immunity, higher the probability or incidence of cancer. So that people think forming a mutant is not a big deal. What the body does is cleaning up the mess. When we don't have the cleaning, the immunity is compromised. That is when the cancers will come about, and that is another view some people do have. I agree with that. It makes sense to me. There is another way of how we see it as cancer as a disease of stemness. If we remember, the whole course is about stem cells, right? We have adult stem cells, for example, which we will also look at, adult stem cells. Let me see if we have a space. Okay. Adult stem cells are there, which will keep on producing some number of some cells, which we call it as transit amplifying cells. It has two properties. One of it is self-renewal, and other one is differentiation, right? Assume this is a little bit of differentiation, and we call it as transit amplifying cells, which will divide rapidly, okay, to form progenitors, and the progenitors eventually will form completely differentiated cells like this. Okay, you can think of it as Diwali. You burn some crackers. You can think of it like that. I should have put it in vertically. We will do that again later. So the stemness is there are some properties here. Self-renewal is a property of the stem cell. Transit amplifier, once a division has made and a differentiation has occurred, these are called as transit amplifying cells. And from here on, they do not have self-renewal. They just keep dividing, and they will eventually divide into terminally differentiated cells. They will work for some days. I mean, they will function for some days, and then they would age because of wear and tear, and they would die out. That is the general scheme for majority of the tissues. So what if some mutation may have caused self-renewal capacity in any of these cells, right? You can see it like this. Somehow the cell is not going to die out. Say, for example, there are two things you should see. If a mutation has occurred in any of these cells, because of which the cell will not divide any further, will gain self-renewal capacity in this stage or in this stage. And there is one more, is that the cell may not die when it is supposed to die. I mean, when there are severe damages during wear and tear, and at one point the cell has inbuilt program to kill itself by, say, apoptosis, and it will die. So if the cell is not dying, or if the cell has gained, because of some mutations, it has gained self-renewal capacity, then that would result in abnormal number of cells, and that is nothing but cancer. Okay, I hope I gave you a perspective. Cancer is a complex disease. You will find some people talking, why are doctors not able to not able to uh, cure cancers, and many things people talk about it. It is not one thing. It is a complex set of diseases which have common point is it is a genetic mutation, and the second thing is it's uncontrollable growth. These two are the common, because we have about 200 or 210 cell types, and many of them can have a mutation, and because of which you can have a cancer in any of these tissues, and each of them is unique in that way. The only common property is that there are mutations and that they are growing uncontrollably. So there are some hallmarks. Now I think people would say there are 8 hallmarks and so on. The first thing, as I said before going further, I would also want to put into your mind a special one of the perspectives in mind, and that is called as the cell number homeostasis. I will just assume that there is a tissue, and based on the size of the body and so on, there's supposed to be 1,000 cells in the tissue to function properly and appropriately. This cell number should be maintained all the time. You can refer it to as cell number homeostasis. And there is a rate of production, and usually that is from adult stem cells. And there is a rate of death because of wear and tear. So if the rate of death increases, the cell number will go down, right? That's not going to help, because you need 1,000 cells for this tissue to function properly. So anytime there is some cells have died, if there are some cells that are dead, the adult stem cells will pitch in and will produce more cells. So this is rate of production. And here is the rate of death, cell death. So there should be a balance between the rate of production and rate of death to maintain this cell number homeostasis. And what decides how many cells should be there in that is based on the functional requirements of that body size, and other factors like that. So if there is any mutation because of which you have increased rate of production, or if there is any mutation that decreased the rate of death, then we would have higher number than required number of cells. That is, you can call it as neoplasia, right? So somehow, if any of the mutations that may happen in the genome, and if they have caused this kind of increase in rate of production of cells or decrease in the rate of cell death, then you would have more cells. So I would want to put in some words into your minds which you may have already heard, and I hope it will be helpful to you. So you know about oncogenes, I guess. Oncogenes are the ones that if there are mutations in the oncogenes, they will increase the rate of cell reproduction, cell division, and those would cause the increase in cell number. There is another one that is called as, there are another set of genes that are called as tumor suppressor genes. That means they usually are involved in cell death, but because of mutation, when the tumor suppressor genes become non-functional. Okay. Now I hope you can try to understand this here. If the tumor suppressor genes become non-functional because of a mutation, then the rate of death would go down. That means the cells that are supposed to die are not dying. That is one problem. And in the oncogene side, it is gain of function. That means maybe there are larger number of genes produced. Maybe the gene that is produced is remaining active all the time or hyperactive. Then it will trigger the rate of production of cells or cell division, and then you can have increased number of cells. Okay, so here are the hallmarks. The cells that are present in the body they are present in a niche, which means they are constantly receiving the cell signals from the other surrounding cells and also the body fluids, hormones and cytokines and so on. And that is important for their growth. When they should divide, all the information is coming from the outside, like here, for example. If the cells become self-sufficient, in the sense because of a mutation, a protein or a product of a gene remains active state all the time, like that of the oncogenes, then it is telling the cell to divide or kind of inducing the cell to divide constantly, and then you are increasing the rate of production, right? Then that is one of the problems. And resistance to anti-growth. As I said, some of the tumor suppressor genes will inhibit normal cells to divide unless they should. And if they become immune to the suppression, that means they have broken the bars and then they just want to divide, there is no more resistance, they have gained resistance against anti-growth signals, then they become irresponsive to the signals of no growth from the outside. And that is one of the problems, like inactivated cell cycle checkpoints. Similarly, immortality, that is cell death pathways, as I said, in this case, where the apoptosis should have happened, somehow it is inactivated, because of which the cells will divide constantly. And this almost same for me, resistance and immortality. This is something interesting. Sustained angiogenesis. So if there is a tumor like this and it was being supplied with blood, tumors are actually growing much more metabolic rate than the normal ones. So they have high energy demands. They need glucose and oxygen in higher concentration. Say these three arteries are supplying it to the tumor. And after a while, the tumor has grown this big. The same arteries cannot supply sufficient amount of energy and oxygen to the grown tumor. So there should somehow be formation of more number of blood vessels, and that formation of blood vessels is called as angiogenesis. So through VEGF pathway, it will increase the number of arteries that will form blood vessels, so that there is more supply of energy and oxygen to the tissue. And then eventually to undergo metastasis. As you can see, this is cancer in a way is a cascade of mutations. How we can see that is, one second. How we should see, putting all the things into perspective, is one mutation happened. Because of that mutation, cell cycle rate is increased, because of which, if the replication is happening more, we might have more mutations, because of which you will have more mutations. Eventually, what you're seeing is a tumor. If you're seeing the cells of it are having multiple mutations all the time. And some of them during those may arise new cells, which may need not be in contact with other cells. They would because of mutations, they might lose the need, and then they may just flow along with the blood into the different parts of the body. And if they come across, if they get settled into some of the body parts, where it is conducive for their growth, they would divide and establish secondary tumors. And that is metastasis. Okay. So these are some of the hallmarks for cancer. And I hope you understood this much. There is not much to understand in these ways, but you should probably build a perspective of how we can fit cancer as a disease with relation to stem cell biology. And that is for one of the reasons why we have it here as well. So there are multiple steps. This is about colon cancer. There are 2 mutations in APC, which will induce some of the cells to grow further, and more mutations will accumulate, and then the tumor starts growing. And with each time, if a cell is changing in its morphology or in the properties or functions or behaviors, it means it is a result of a mutation. So once a mutation happened, that is like a cascade of many more mutations to occur, and eventually it will reach a stage of metastasis where some of the cells will move out into the bloodstream and get circulated into the body. So if you also see the progression in general, and the first place where the tumorigenesis happened, or some cell has transformed, a normal cell has transformed into a tumor cells. Here we should use the word transformed. That means a normal cell became cancerous. And there you will have abnormal growth, and that point is called as the primary tumor. And then with increased angiogenesis and further proliferation, it will increase in size and metastasis would occur, meaning that some of the cells would lose the cell-cell connections and it will enter into the bloodstream and get circulated into the blood, through the blood to different parts of the body, through the heart, and then it would reach into certain organs. One of the organ is lung, and there they might settle into some cells and establish a microenvironment for their own growth, and they will proliferate and form secondary tumors in situations like lungs. What does a tumor need to grow? It would, of course, need space, energy, and oxygen, right? What else is a better place than lungs? Right? Lung is a good place. Not that every tumor will, every type of secondary tumors will form in lung or only in lung. There are different types of cancers. Some of them, secondary tumors, could be found in the brain, many of them on the bone, and many of them in the lung as well as a secondary site. I would just try to rationalize why a lung is a good place. To start with, lung is a cavity. It has a lot of space there, and it is directly accessible to oxygen, and it is constantly supplied with blood because that is where oxygenation would occur. So lung is a good place because all these the space, energy, and oxygen are available there, and that will be a good place to proliferate. As I said again, it's not that all types of tumors can form or will form secondary tumors in the lungs. Okay. So there are some properties that are common between stem cells and cancers. Okay, so the behavior wise and the signaling pathways are almost similar. We will read about these signaling pathways, so I wouldn't touch about at least these 3. We will read about them: Wnt signaling, hedgehog signaling, Notch signaling, and FGF signaling. If you see what I meant by the similarities between the stem cells and the tumors, is that Wnt signaling, for example, is involved in the maintenance of these cells or adult stem cells of the gut, hematopoietic or epidermal ones. And it is also responsible or is dysregulated, because of which colon cancer and epidermal tumors may occur. Similarly, this is sonic hedgehog. In hepatopoietic, these stem cells will are hedgehog is involved in the regulation of these cell cycles, whereas it also can be involved in basal cell carcinoma and so on, and not signaling as well. So you find there is a behaviorally there is stemness in cancers. That means self-renewal and ability to grow. The difference between a stem cell and a tumorous cell is that stem cell will only grow when it is required to, will divide only when it is required to, whereas cancer cell does not have that regulation. Okay, but otherwise both of them have kind of stemness, if you can try to call it that way. So there are two general models, and again I will give you one more reason why we should why we are studying about cancers in stem cell biology. If we take any of the cancers and we take the cells of the cancer from one tumor, for example, it's not that they all are having only one cell type. In fact, they have more than 3 or 4 cell types. Okay, that means the cancer cells in a tumor are heterogeneous. They are of varied types. So why is it so, and how do they arise? Is it that if I take a cell type and put it into some other location, we will discuss about the characterization techniques and other things slowly. This is an introduction to cancers and cancer stem cells. Is it that A will give rise to more of A cells, or is it that A can give rise to all the cell types? What we know for sure is that the heterogeneity of tumor. Now we have to prove is it that A can give rise to only A cell types and B can give rise to B cell types, C to C, and so on? Or is it that the cell type A can give rise to all the other cell types? Okay. And if we remember the stem cell models in general, adult stem cells, as I was describing before, adult stem cell will give rise to transit amplifying cells and eventually give rise to differentiated cells. They have a potency. Assume it is oligopotency or multipotent. Okay. This adult stem cell might give rise to about 6 or 7 cell types. Right? That is how an adult stem cell or tissue systems are usually organized. So some people think that this is the model I was talking. For cancers too, it is the same thing. There are a small subset of cells that are responsible for growing the whole tumor, all the cell types. Okay, these cells, say BCD for example, they cannot give rise to tumor. Only A cell type can give rise to a tumor. That is the model of cell, what is meant by the cancer stem cell model. So here it is, the description of the model. I'll just take this and one more slide and I will stop. Stochastic model and the cancer stem cell model. In the stochastic model, you can take any type of cells. Here there are 3 different cell types. You can take any of them and all of them can give rise to a tumor. Stochastic model. In the cancer stem cell model, although there are say 1, 2, 3, and 4 cell types, it is only majority of them cannot give rise to tumor, but only a small subset of them can actually are the responsible ones for maintaining or the aggressive growth of the cells or the tumors. And those are referred to as the stem cells of the cancer or cancer stem cells. Okay. If you look at the cancer stem cells and normal stem cells, I think this is the cancer stem cells and these are the embryonic stem cells. They have similar properties also in their morphology. They are small and they are mostly quiescent. They do not divide. and then they divide, there would be rapid burst of cell death, cell proliferation, like in the transit amplifying cells. And why we are learning this is the last slide I will take, and I will repeat again in the next class. So if there is a person and the person has a tumor somewhere, and we know the technique or the therapeutic agents we will use are through chemical chemotherapy or radiotherapy, and sometimes a combination of both. And we also have the other one, it is immunotherapy. These are the three broad categories of how we try to treat cancers. So because of these chemotherapy, radiotherapy, and immunotherapy, the person, individual, majority of the times gets well. And if we wait for some years, the tumor comes back. And it might be in other locations as well. And again, treatment, and then eventually death. So these are not easy ways, chemotherapy, radiotherapy. They're actually very tough on the individual suffering from the cancer. And the point that I'm trying to get here is, when we have treated it, how is it that it is coming back? That's the question. Why is it relapsing? That is one of the reasons why we have very few therapeutic ways or complete curing. Majority of the cases, some of the cancers can be cured, but not all. Some will just prolong the time of survival, although the survival time could be actually troubled because it's filled with so much of very painful therapeutics. So what the assumed way is, we are trying to, when we kill, we are using chemical or radio. For example, if we are using chemical or radio, most of them are DNA breaking ones. If there is a DNA, the genome is broken, and if the cell is constantly just dividing anyway, then it is not going to break. If a cell with broken DNA is divides, it will form two non-productive cells, and they are useless, and then both the cells will die out. So that is general strategy for radiotherapy and chemotherapy. So what we are doing actually probably is that when we add this chemotherapy or radiotherapy, we are killing the cells that are growing rapidly, the rapidly growing ones. And the cancer stem cells apparently are very slow growing, whereas the other cells are the rapidly dividing ones, but they would die out, like in the adult stem model that we have discussed. So when we use chemotherapy or radiotherapy, these cells will die out, but the slow growing ones will survive. Why is it? If a mutation occurred like this, in this case, because it is slowly dividing, it has time to repair all the damages. Right? First of all, it might not be affected as much either. And even if it did, there are repair mechanisms. There is time sufficient for repair mechanisms, and then it will divide only when it should. So the cancer stem cells apparently survive, and the rest of them die out. And so after a few months or years, the tumor relapse. The cancer stem cell would establish a new tumor. So the goal or why, the objective of why we are studying this is somehow develop some ways of trying to kill specifically the cancer stem cells. If we have targeted the cancer stem cells and and killed them, and then we would have the tumor, the rest of the cells will die out eventually. They do not have the capacity to grow and keep on growing. They will divide for a small burst of cell divisions, and then they would wear and tear and die. So if we have killed all the cancer stem cells in this case, then we would have probably the rest of the cells would die and tumor degenerates, and hopefully there will not be any relapse of the cancer. And that is the reason why we are studying this. as a part of cancer stem cells and stem cell biology. And it is also a good way to build a motivation to why we should be studying all these things, right? I'll stop here. If you have any questions, you're free to ask, but otherwise I will repeat a few things maybe from this slide on in the next class too. Any questions? I'll switch off the recording. Please tell me if you have